Thank you for inviting me. I'm Bernhard from the University of Bern, Switzerland. I'm a biologist, and I'm here because I have a replication problem. I'm also the last person standing between you and the coffee break. A few years ago, two studies, one conducted by researchers of the company Bayer and the other one by researchers of the company Amgen, shocked the world of biomedical research. In the first study, published in Nature Reviews Drug Discovery by Prince and colleagues, the researchers reported outcomes from 67 in-house replications of original studies published in academic journals. And with replication studies, we really mean full replication, starting with newly synthesizing the compounds, applying to animal models, measuring the things and analyzing the data. So it's not only reanalysis, but complete replication. Only in 24 studies, that's 36%, could they fully or partly reproduce the original findings. In a similar attempt, researchers of the company Amgen tried to replicate 53 what they called landmark studies in oncology, many of them published in high-impact journals. As they deliberately focused on sensational findings, they expected that not everything will hold up, but they were still surprised that only in 6 out of these 53 cases, they could confirm the claims of the original study. That's just 11%. So this was really a surprise. A few years later, there was a review by the Journal of Nature, most of you will know this, where the journal made a survey asking 1,500 scientists whether it ever happened to them that they could not reproduce an outcome of another group, of a colleague, or of one of their previous own studies. Many of them said yes. Furthermore, Bernhard Baker asked those researchers whether they thought that there is a reproducibility crisis in science, and 52% said yes, a significant crisis. Another 38% said yes, some sort of crisis, and only 10% were not concerned at all. Now, yesterday we had a short discussion whether we should still speak of reproducibility crisis, given that there is an audience of 400 enlightened scientists in this audience, which is already beyond just recognizing this crisis, and we are already on the way a step further. But at the same time, if you look at biomedical research, there are about 100,000 researchers all over the world which do business as usual and have not changed their method and still produce often irreproducible studies at an hourly rate of almost. So I would say we still have a reproducibility crisis. In this survey, Nature asked the researchers also what they think the main reasons for poor reproducibility are, and also what the solutions could be. I won't go to all these points, but it's basically ranging from selective reporting and publication bias, basically down to outright fraud. One thing which is very clear is that all researchers agree there's not a single cause for poor reproducibility, but that there are many different factors contributing to it. And there's also another telling thing here, but I will come back to that in a minute. If we now think of preclinical research, this works as follows. You plan a study with a study design, you produce the reagents, you apply them to an animal model, you take some measurements, you analyze the measurements, and then to publish it. Easy as that. Well, in reality, it's not always that easy, but that's the ideal case. Nice workflow. Now, when we look at this list of proclaimed reasons for poor reproducibility, what we see basically is that they apply to all to most of these stages in this research pipeline. So what is mentioned is poor experimental design, which is fall in the planning stage. Low quality reagents, measurement error, errors in the statistical analysis, and then of course publication bias and so on. So all these different steps might be problematic, and yeah, basically what you do at all these steps can contribute to reproducibility. Strikingly, there's one step in this pipeline missing, which is not mentioned in the list at all, and that's the animal model. And this is surprising because there are at least two really critical things concerning the animal model, and the one is the validity of the animal model, and the other is the variation in the animal model. Now, there has been a heated or is an ongoing debate about the validity of animal models for specific human conditions. And very likely, this sometimes missing validity is a reason specifically for the translation failure between preclinical animal studies and then clinical human studies. The second point is the variation in the animal model, and this is what I will talk about for the rest of this presentation. So, lab mouse does not equal lab mouse. Two mice might differ in their fur color, as you can see here. They might differ in body weight, but they will also differ in many other aspects, in physiological parameters, in immune responses, in responses to treatments and drugs. All of these different properties of the organism together constitute the phenotype of the mouse. So these phenotypes differ, and the question is, where do these differences come from? Now, there are three main sources. There are genetic differences. Mice are not all the same. Lab mice are different. In dogs, we have these different breeds. In mice, we have also different breeding lines, which we call strains. In contrast to dogs, they have much more fancy names, like C57 Black 6J or DBA. And these breeding lines differ genetically. But even within these strains, we have genetic differences. They are not clones, so there is genetic variability within strains. And sex is another source for genetic variation. Male and female mice differ in a complete chromosome. Apart from genes, there are environmental influences that also shape the phenotype. How much food an organism gets influences its growth, its body composition, its health status, but also many other stimulus like early stress during early phases of the organism, or cognitive stimulation, or basically every exposure to the environment that an organism experiences can influence the adult phenotype. And finally, there is something which is called G-by interaction, standing for gene-by-environment interaction, basically meaning that how an organism responds to environmental input depends very much on the phenotype. So on the genotype, sorry. So one genotype will respond to environmental stimuli in this direction, and another one differently. The relevance of all these sources of phenotype variation for day-to-day lab mouse research has been nicely demonstrated in a study already 20 years ago. It was a study by Crabb and colleagues who made a big replication study. So they took lab mice, 8 different strains of mice, both males and females, and made a whole battery of standard tests like the open field test, a plus mace test, water mace test, response to cocaine and alcohol, and some other things. And they replicated all this in 3 different laboratories. They went, as they say, to extraordinary lengths to equate test apparatus, testing protocols, and all possible features of animal husbandry. The variables they equated across laboratories included the apparatus, testing protocols, SOPs, of course, the age of the mice, the housing conditions, and so on and so on. So they really tried to standardize between these 3 labs. I will just present results for one of these tests. This is the elevated plus mace. It's a test frequently used in psychopharmacological research, where you have basically a T mace with two closed arms with walls on the left and the right side, and then you have two open arms. You're placing a mouse in the middle, and then you're just observing the behavior. If the mouse is spending all the time in the closed arms, it's considered to be very anxious, while when it's also venturing into the open arms, this is considered as exploratory behavior and a sign of reduced anxiety. So this is very often used to test the anxiolytic or anxiogenic effect of drugs. Now, if we look at this data, what we see are strain differences. So if we just focus on the first two strains, we see this, that, okay, sorry. We see these two strains, HA and C57 Black 6, and you see that the Black 6 show, even though there's a bit difference between the three labs, so it's Portland, Edmonton, and Albany, there's some variation, but overall, the Black 6 are much more active. They're more often going into the open arms, so they are less anxious than the HA mice. So this is a genetic difference. But apart from these genetic differences, we also see environmental differences. Despite standardization in all 8 strains and also in both sexes, we see that in Edmonton, the animals were less anxious than in Albany, for example. And finally, we also see gene by environment interactions. Just focusing on these last 2 strains, what we see here, that the last strain, the B62F2, was in Albany much less anxious than the 5HT strain, but in Portland it was exactly the other way around. And in Edmonton we didn't see any difference at all. So what do we make out of all these differences and how can we explain them? Here, the norm of reaction, an evolutionary concept, might help us. The norm of reaction is a concept introduced by the German physiologist Richard Wolterek over 100 years ago. And it can be basically described as a function describing the relationship between an environmental parameter and the expected trait value for a specific trait for one genotype. So if we have a certain environment and then animals experience in a certain environment, this might influence somehow the trait value. So if you have a specific environment, let's say environment 1, so genotype growing up in environment 1, we would expect that it shows then a certain expected trait value, where in biomedical research, trait value usually means a response to a specific treatment or compound. Of course, there's around this expected value, there's some variation due to other reasons, random noise and so on. But if the same organism would have been raised in a different environment, environment X2, we would have expected a different trait value, so a slightly or largely different response to a certain treatment. And the reaction norm is basically the function describing this relationship between the expected value and the environmental parameter. This reaction norm can take on any form, and also it can be different for any genotype. So if you have a different genotype, like this one here, this might have a completely different reaction norm. And what makes matters worse, these reaction norms are actually not functions in two-dimensional space, but environments have many, many parameters, and basically you should imagine it as n-dimensional hyperplane in a high-dimensional vector space. But for the sake of simplicity, graphical representation, I will stick to the two-dimensional representation. Now, how does the norm of reaction can help us to improve biomedical research? Think of a simple study where we just rear animals in one environment, then we get some expected trait value in lab 0. Now, if researchers in a different lab try to replicate this study, they will basically raise their mice in a different lab, which has different conditions. Labs always differ in the environmental conditions, the temperature, the humidity, but also things like the odor in the lab, the background noise, stock intensity, the mood and the temperament of the animal caretaker. These are all environmental parameters that can be relevant for the mice and which differ between labs. Some of them, if you want, you could standardize, but others really defy standardization. You will always have differences between labs. And this may cause that in this other lab, you get another trait, and you should expect actually another trait value. But what happens is if people in this lab just replicate this study and they get a different finding, then they report we could not replicate the original finding, period. And then this is seen as a replication failure. Now, let's take a different approach. Yep. Instead of making our initial study in one lab, let's make a multi-lab study with 5 contributing laboratories. So the same experiment is done in 5 different labs, and what we get is 5 labs which differ in some environmental parameters, and therefore we get different expected values. And if we get data from all these different labs, what we get in the end, and just pool them, what we get in the end is a much wider distribution. And if now somebody is coming and making another replication study in just one lab, lab 7, for example, might be this outcome, what we see is that the mean value that you get out from this study might still not be exactly the same as what we got in our original multi-lab study, but the general overlap of these distribution of the frequencies of the observed values is small enough that we say, okay, there is definitely not a real contradiction in there. We can call this successful replication to some extent. And if this replication study would also have been a multi-lab study, in fact, in most cases, we would see a very large overlap between the distribution of observed values, and we would call it successful replication. Now, one can come and say, that's a cheap trick that you're doing. You're just broadening your confidence intervals, and then it's easier that outcomes replicate. Well, technically, that's true. That's what I'm suggesting. But the important thing is that I suggest to do this because it reflects biological reality. Individuals differ. Individuals differ because of genetic differences, but they also differ because of environmental differences and gene-by-environmental interactions. If we get away with all the environmental variation, we are sampling from a very reduced sample space. We are not acknowledging the full diversity of individuals which is out there. Sociologists and psychologists know for a long time this is a bad thing to do, but in biomedical research, this is basically standard practice. Okay, and this problem that we standardize so much, and this, I mean, in the worst case, could happen that we just make an experiment in a lab which has very extreme environmental conditions, and due to the reaction on this leads then to very peculiar expected values for specific traits. And if we just have one lab, it could be that we standardize to a very peculiar and strange situation. And if we do this and then we try to replicate, it becomes very difficult. We will never find the same result again. And that's called the standardization fallacy. The term is attributed to Bernhard Voekl, but actually Ronald Fisher in his 1930 book on experimental design already said the same thing. Basically, that excessive standardization of every aspect of the study subject will lead to idiosyncratic results that hold only for this specific lab, but that cannot be reproduced under even slightly different conditions. And this is, I believe, what is happening in mouse research today. If we make a study just in one single lab, which is highly standardized, and the problem is that standardization within a lab is usually taught as good scientific practice. But this is exactly what might lead to reproducibility problems. And the suggested solution is to conduct multi-lab designs to basically diversify your study samples. Whether this could really rescue reproducibility in biomedical research is a question we try to address with a simulation study. So the theoretical argument in favor of multi-lab studies is quite convincing, but the question is, does this really matter for the kind of data we have, and how much effort do we have to put into this in order to make it work? So we investigated this by making use of the Camarades database, thanks Malcolm, and we selected 14 different treatments, interaction studies, where we found in the database quite a lot of different studies. For example, for hypothermia treatment of stroke, we found almost 100 studies, mouse studies, where this treatment was applied. The reason why we have so many studies there is that hypothermia is one of the few treatments that works very well in the clinic. So whenever a new drug for stroke treatment is researched for, hypothermia is included in the study as a positive control. And what we did is we took those studies and then we generated new simulated new studies by sampling from the reported distribution as they were, from the distributions as they were reported in the original publications. So in one case, we randomly picked one of those publications, looked at the reported means and standard deviations for the control and treatment group, sampled from those distributions, and created new values. So 12 animals for the control group and 12 animals for the treatment group. And then we got some difference, and then we could basically calculate the effect size and whether we found a difference. In the other case, we made the same, but we didn't sample from one distribution each, but we took 3 papers and asked the question, what would have happened if the authors of these original study would not have done these studies on their own, but would have joined forces and tried to make a multi-lab study together, pooling the data and making one analysis. And then you get these outcomes. Yes. And what we basically found is that basically what we expected is if you go from a one lab study, if you just have one lab, like here, you have a lot of variation between individual simulated studies. But if you go to multi-lab designs where you have 2, 3, or 4 labs, you see 2 effects. So on average, the within-lab variation increases a bit. This is this broader confidence interval. But on the other side, the between-lab variation decreases, which is indicated by this smaller dance around the estimate of the meta-analytic mean here. So between labs, we don't see so much variation. We, as a measure for reproducibility, we calculated the coverage probability, which is basically the probability that the confidence interval of one study would capture the estimated overall estimated effect size from the meta-analysis over all studies in the study pool. If we have just one lab, this coverage probability is relatively low, going from 0.38 to 0.6. But as soon as we have a multi-lab setting, it reaches 0.8 or even going beyond 0.8, also depending a bit on the sample size. Now, one important point is that in these 2 studies, the overall sample size is exactly the same. So we have 24 animals here, we have 24 animals here. So every effect that we see, or any difference that we see between single lab study and multi-lab study is not an effect of increased sample size, but is an effect of increased variation within our study. And actually, I think this is rather good news. So 2 things that you see is that you can improve the coverage probability, which is a measure for reproducibility, with just having a few labs in a study. So the number of labs in a multi-lab study that you need to get really a big improvement is not prohibitively large. 3 labs will be usually fine. And in fact, the biggest improvement you can already see when you go from a single lab study to a twin lab study, basically confirming the old wisdom that every result should be replicated independently at least once. That's exactly what we see here, what we gain from this. And the second thing is the second good news from an ethical point of view is that we can get this improvement without needing more animals for research, because this is, of course, also an ethical issue. Okay, so I'm already close to summarizing everything. So a single experiment will never capture the full diversity in traits and responses to treatments existing in any animal model. Highly standardized single laboratory studies are therefore a source of poor reproducibility, that's the standardization fallacy, because they ignore biological important variation. If we just have one single environment, this means that we are sampling from just one point on the reaction norm, and we're just ignoring that there is diversity in phenotypes out there, and diversity that matters. And multi-laboratory studies and other ways of creating more diverse study samples provide effective means to improve reproducibility in biomedical research. Here, I just focused on the multi-laboratory studies as a way to increase the diversity within a sample. But another way to go would be that we have one lab, but we consciously increase diversity in this one lab. There are different ways, of course. One way is to increase genetic diversity. What is usually already done is having both male and female mice. What some people also advocate is using either outbred strains or different strains. But another way to increase diversity within a lab is also to basically increase environmental diversity, change some parameters in the environmental conditions. This all can help to increase the overall diversity of the phenotype, because in the end, it's not the genetic diversity, but the diversity of the adult phenotype, which is relevant for the response and how we interpret this response. And finally, of course, the statement, we have a moral obligation to prevent wasting animals and resources for non-reproducible and hence inconclusive research. And this is actually where I'm coming from. So at the University of Bern, I'm at the Animal Welfare Department, and we are interested in reproducibility of biomedical research mainly because if this research is not reproducible, it is, of course, a waste of valuable resources, funding resources, and there are estimates that it's several billion per year worldwide, but it's also a waste of animal material, and we have a moral obligation. We can use animals for research if we can gain knowledge from this research, but we have a moral obligation that we really gain something from this. Otherwise, we're wasting animals for nothing, and this is unacceptable, basically. Okay, so thank you very much. Yep.
Is there any way to tell whether the Amgen or Bayer studies would have given very different results or even different results had they been multi-lab?
No, we don't. We can't say that. And actually, as far as I know, the raw data of those studies not yet really published. So what was published was basically a summary of the outcome, but I haven't seen the raw data of those studies.
In the Amgen study, it was the labs participated, the original labs participated on the condition of anonymity, and the data will never be available. Mm-hmm. I don't know about the Bayer.
Hi, Tim Arrington, COS. So I like this. I've always liked this idea. Pharma companies are actually starting to do some of this, especially the latter one that you mentioned, which is inverting it, not focusing on a single strain, but eventually getting into all relevant disease models, because you're essentially trying to get closer to generalizability. So very similar to what we do in clinical trials, which is you scale up. And that's actually my question, which is, this is quite labor intensive. I think it's a great thing to do, but I'm just curious about your thoughts of when to do it. So it takes a lot more resources to coordinate multiple labs, just like it takes a lot more resources to maintain and conduct the research across multiple strains of mice. So do you envision, would your suggestion be all of it needs to be done this way, or that you layer, right? That you start with a single lab, and then if you want to, you basically say, start to layer on these approaches instead of just kind of going all in on multi-lab or multi-strain approaches?
We actually advocate the idea to start very early on. And this is something where, we do not make many friends because, as you say, it's it's a lot of more administrative work at the at the start, and also more work. I mean, concerning the resources, we're speaking of making a multi-lab studies which uses exactly the same overall number of animals than a single lab study would use. So we don't need more animals, we don't have more costs. Of course, transportation costs might increase a bit. And of course, setting up the experiment in several labs is more expensive. But on the other side, if you do it in one lab, and then you replicate it in a different lab and it doesn't replicate, it's also a lot of money. And then you have basically two studies and you don't know much more either. So even so, the single study becomes more expensive. We think that we can, this suggests that we can reduce the replication failure. That we can increase replication success by up to 42% points, which means we have far fewer studies which don't replicate, and this is also where we can save a lot of money. And the other thing is, while it is true that planning a multi-lab study is very tedious, on the other side, it also means that you usually have better protocols because you discuss with your colleagues. It's the same as with pre-registration. It's a lot of work at the beginning, but we hope that it pays off because you get more reliable, more reproducible results, more reproducible study protocols. And the same is with multi-lab studies because there are 3 or 4 researchers sitting together trying to really figure out, okay, how do we do it? What do we do? And basically researchers, colleagues checking each other and agreeing on a common protocol, having a lot of communication about how to do this. And this all helps additionally to the statistical argument that you increase the percentage of replication, but that too will additionally boost a reproducibility of such studies.
Hi, my name is Tom Trainer. I worked 10 years in industrial biotechnology, R&D. I'm now an independent consultant. So was that presentation or that paper from 1999 telling me that if I want to relieve the anxiety of the last couple years, I should move to Edmonton or anywhere in Canada? Okay.
Yes, I mean, it was 8 different strains and both sexes. I just showed data from one sex, but in the other it was the same. So in Edmonton, mice were less anxious in all 8 labs.
All right,
so that was really consistent.
Yeah, my real question. So the fallacy of standardization I've observed is widespread in industry. A lot of graduating PhDs, master's degrees, they come out saying, I know how to design really good experiments. I control everything. But then you end up reading in the papers about, this company failed to scale up their protocol or whatever. And it's basically the fallacy of standardization. They weren't designing the research program to cover enough of the variable inputs that were going to be experienced as the technology moves to other sites and situations. So my question is, and now another challenge, of course, though, is that when you broaden those confidence limits, there's a reasonable expectation we're going to have a lot more negative results. The studies that seem to be sufficiently powered when the standard deviations were smaller are no longer sufficiently powered for the same number of replicates, unless you have this really fortunate situation where the standard deviations in the different sites are the same. You mature your statistical analysis to a multivariate analysis. Anyway, question is, what tools, software or otherwise, do you think would facilitate more of these important multi-lab experiments? Is it just a matter of popularizing the standardization fallacy, or do we need it to be easier to publish negative results, or what would you suggest?
I mean, statistically, multi-lab designs are not really an enigma. You can basically, it's a two-way ANOVA design. You can analyze it as a blocked ANOVA, or you can analyze it as a mixed effect models, having lab as a random factor, as you prefer, or sometimes you can even analyze it just pooling the data, but it's a contentious issue, and I don't want to go into this. But in principle, the statistical tools to analyze a multi-lab studies are here, and they're not really complicated. And every researcher, every PhD student, every statistician is able to do that. So we do not really need new tools to analyze this. I think how to promote this is basically a question, again, of research policy. I mean, this idea that you need an independent replicate, it's on the very basis of modern science. It was in the 17th century at the Royal Society in London. They said, we just believe in the result if it is at least replicated once in England. And so, and also today we basically say we want replicates. We want to see a result not only in one result, but we want to see replicates. But what happened is basically that on some reasoning, people said, okay, my replicates are the animals that I have in my lab, but they are not independent replicates. But they are often sold as independent replicates. Independent replicates means that I have animals that experience slightly different conditions. An independent replicate means that we replicate everything which is necessary to really see the effect we want to see, but not more. If we also basically standardize all these other things, sometimes consciously, sometimes unconsciously, we do not know what else we put into the system, that what we do not consider, but which might affect the results. So this idea that my 10 mice in these 5 cages in my lab, that they are replicates, I think this is a basic problem. And basically the importance of replication is fully acknowledged, but there is no journal or there are hardly any journals which really require that you can prove that you have an independent replicate of your study. If you submit to Nature, you can submit one study. You don't have to say somebody else replicated this or we replicated this a second time. Same for funders. They do not, even so there are always these statements, replication is important, but you can still submit funding proposals where you just say, okay, I make this and this study, without saying, okay, this and this person will replicate, has to replicate, or we will replicate it a year later or so. So if replication is taken really serious, I think this would really help.
So since we're coming right to the end of our time, I'm going to exercise the chair's discretion in the last 30 seconds to ask you one final very quick question to forecast a theme that's going to come up on Sunday morning at a panel at which I'm quite confident things will be said which will cause most people in the room to stop breathing temporarily. So here's the question. One thing I think you haven't mentioned about any of these mice studies is random assignment to condition and the experiments. So does your lab randomly assign mice to treatment or control?
Yes.
You do? Okay.
Yeah, but I think that's very common.
We'll be talking about that on Sunday. Thank you very much. Okay, on that note, coffee break. Thank you.